Hello, and I want to welcome you all today as I start off this session. First, I want to give you a quick idea of what to expect in this session. I am one of the BioRED track organizers, and myself, Wei, and Puting will cover the overview of an intensive work that has taken the most of our time for the last 6 months. Of course, we will make it all sound easy to you and show how wonderful everything is. Well, all is well. That ends well. We will talk about, why we do this track and how this relates to the to the research cycle as a whole. We will talk about the dataset. We will talk about the track participation, the evaluation, and we will discuss successful approaches. But first, let me start about what is BioRED and a little bit on the BioRED history. BioRED actually started in 2021, and at the time, with the COVID restrictions, a few people from our team, in collaboration with 4 curators, developed the annotation guidelines and selected 600 articles from publicly available corpora reaching genes, diseases, chemicals, and other biomedical entities, and annotated them with relations, relation types, and novelty. These 600 articles were used for the Litecoin NLP task, which attracted a lot of attention. Now, in late 2022 and early 2023, as we started planning BioCreative VIII, the question that we raised was, can we build relation extraction models that can actually work for new and previously unseen articles? So we gathered a group of excellent curators at the National Library of Medicine. They have an average of 25 years of experience in indexing and curating biomedical articles with high degrees in chemistry, biology, medicine. And we curated another 400 articles, which constituted the dataset for this track. So we have, for BioCreative VIII BioRED task, the dataset contains 1,000 PubMed articles, fully annotated, with entities and relations. But why should we do this? Why do we need a challenge for this? I would like to take a bird's eye view together with all of you and to look at the biomedical information extraction research as a whole. So what does it look like to an outside user? What do they input? What do they expect to retrieve in return? And what kind of processing needs to happen within the knowledge space so that we may accurately return results? A reasonable scenario is a health researcher asking a question similar to what we illustrate here on the left side. T2D therapeutics candidates. And basically, the search engine needs to deduce that T2D stands for type 2 diabetes. In this context, it is a disease, and we are looking for knowledge that is extracted from all the vast amount of publications that we have, that is backed up by experimental evidence, that there is a relation between this disease and certain drugs and chemicals that could be therapeutics for this disease. Now, let me take you to the right side of this image and see what's shown there. Here we have depicted the accumulated knowledge of named entity recognition research in biomedical NLP and the hundreds of hours of human curator time that has been spent to record this information in the knowledge resources. We have catalogs of genes, proteins, species, diseases, drugs, chemicals, genes, variations, and so on. When we consider this large context, the relation extraction is in the middle. It's what needs to happen so that we may satisfy the user requests and with it adapt to the new technological advantages of our. So that was the big picture. This slide documents that in PubMed, the collection of all literature that pertains to health and medicine, when we look at all the user queries, we see that certain entities appear very frequently, which also brought forward all the research on named entity recognition for these entities. On the right-hand side, we see a schematic of how these entities relate to each other based on what we see in the PubMed articles. Of course, relation extraction is not new. It has been addressed in many research challenges. And with this slide, I give you a little bit of a history from all BioCreative challenges. So let's go a little bit down memory lane and let's start from the beginning. The first BioCreative happened in 2004, and it was focused on gene name identification. After that, actually, every single BioCreative challenge has addressed some form of relation extraction. And so BioCreative 2, BioCreative 2.5, BioCreative 3 addressed protein-protein interactions. In BioCreative 4, we have an attempt to do 3 types of relation extractions. We have chemical drug, chemical disease, and drug disease. In BioCreative 5, we have a simplified form of this same track, but only looked at chemical disease interactions. In BioCreative VI, we have 2 relation extraction tasks. We have a chemical protein task and a protein-protein interaction task, but so that they depend on mutations. So it's only when a mutation triggers a protein-protein interaction. And this was discussed within the context of personal medicine. In BioCreative 7, we have a repeat, a continuation of the chemical protein relations. And in BioCreative 8, we are attempting something much larger that has not been attempted before, not only in the BioCreatives, but also in other challenges that have looked at relation extraction. We are looking at a comprehensive relation extractions that we have many types of relations. We're still looking at binary relations, but we're also not looking at relations that appear within the same sentence, but can consider the whole abstract, the whole paper, the document as a whole. So the relation extraction task that we are looking for is that given a document, and for our purposes, we are looking at PubMed abstracts. We provide gold standard entities. We provide their spans, their annotations, their concept types, and their IDs. And we ask the participants to identify which pair of entities are in a relation, are related to each other according to the context of this document, what type of relation they are in, and whether this relation is novel or not. Manual curation considers the whole document. So this is different from previous tasks that only looked at one sentence. The novelty in relations is a little bit interesting, and it's also new to this track because we distinguish between novel findings that are shown as the contribution of the paper, of the abstract that we're looking at, versus previously known background knowledge. View an example. For example, this is a document we are looking at, but let's focus in, let's zoom in at this sentence, and if we actually sort of understand each of the pairs and what are the relations. A novelty is between this variant and this disease. That's the novel relation that is described in this article. The other ones are all background knowledge. So basically, just to repeat, but with a little bit more visual cues, here is a screenshot of a PubMed article. We have highlighted the entities. The types of entities that we're looking at are diseases, genes, or proteins. We do not distinguish between the two. Chemicals, species, cell lines, and variants. And we're going to look for disease pairs between these types of entities. And we're not looking at every single possible pair between those 6, because according to our statistical analysis of the whole PubMed data, the other pairs are not as interesting. Then we have identified these relation types. We have positive correlation, negative correlation, association. Most of the relations are association, and novelty.
Unlike the training set, which was sampled from a list of existing datasets, such like BC5-CDR and NM gene corpora, the test set was extracted from new journal articles published between September 2022 and March 2023. Since one of the goals of BioRED track is to foster the development of algorithms that can handle new, previously unseen literature, such like the COVID-related paper during the pandemic. That is why we are using new articles. We have 8 curators from NM indexing team worked on the test set annotation, which is 2 times of the number of curators in training set. Curators follow the same annotation guideline as the training set. Curators first annotated all entities. To improve the curation speed and quality, we apply PubTailor to pre-annotate the entities. As illustrated in the figure, training and testing datasets has a similar entity composition. To maintain data compatibility, it is critical to make the entity composition and distribution as closely as possible to that of the training set in terms of both entity dimensions and concept identifiers. To maintain data compatibility, it is critical to make the entity composition and distribution as closely as possible to that of the training set in terms of both entity dimensions and concept identifiers. The numbers demonstrate that the entity distribution between training and testing sets are very similar. However, you may notice that the numbers of genes and variants are slightly larger. This was intentional because we want to have more relations involving variants since they were not very well represented in the training set. Finally, we want to demonstrate the richness of the BioRED track corpus for each entity type when we compare the entities found in the training and testing sets. We have a reasonable overlap, as well as new, previously unseen entities. This is expected since we are using new published articles. Here are the overlap cases of the chemicals and diseases, shows about 20% overlapping of the entities. Taken together, the two sets complement each other and contribute to a richer, complete dataset. After entity annotation, curators curated the relations. Here you see illustrated the distribution of the annotated relations in entity pairs, relation types, and novelty. Overall, the relation distribution numbers are compatible with the training dataset. The BioRED track is divided into 2 subtasks. Subtask 1 asks the participant to use the title, abstract, and the human LT entities of given document to extract the relations between entities and their types and novelty. Subtask 2 asks the participant to extract the relations. However, no entity annotations are given. So essentially, the participant need to develop an end-to-end system, which means participants have to develop their own method for entity recognition. Here is one example article. On the left, we see the article title and abstract, where we have highlighted all entities. This article contains disease, gene, chemical, species, and gene variant. The table listed all the relations that were annotated by our curators for this article. As one of these relations is marked as novel because it is described in these articles as new finding. So that is the challenge. Given an article, identify how the mentioned entities relate semantically and make things even more difficult. Sometimes there are entities or more which are related simultaneously, but we are not testing that in this work. You will only look at the binary relations. Now I will pass to Boting, who will cover the track participation and evaluation results.
Thank you, Rezarta and Zhixuan. This figure shows participants from 7 different countries and regions: Australia, China, Japan, Portugal, Russia, Taiwan, and the United States. 14 different teams participate in subtask 1, 9 teams participate in subtask 2. The team submitted 37 and 29 official rounds for both subtasks. And they also continued their work and submitted additional rounds, which we called unofficials, because they were submitted after the deadline. This workcraft summarizes the methods and the resources used by all participants. We noticed that there is a universal use of the bird model, and everybody used the bird. And then among all the bird models, the palmy bird was the most preferred model. And for subtask 2, since the participants did not have enough time to develop their in-house NER systems, we noticed that the majority relies on Puppeteer API to retrieve the predicted entity and their ID. And NLTK species were most popular for data preprocessing. In addition, we see additional resources and tools such as Crafter, all-in-one NER, NCBI interest, or means. Some systems also incorporate GPT models. This table shows the team's evaluation of subtask 1. For each team, we pick their best rounds on their F-scores for predicting a relation pair between 2 entities. Later, teams asked for more times, and we received a number of additional submissions. But in the interest of times, we are not displaying that here. However, we did notice that almost all of them that submitted unofficial rounds improved their F-scores. This means that if everybody had more times, we would have seen even higher F-scores. But let's discuss this table. The first thing to notice that is that the results are very similar. A reason for this similarity could also be the similarity in their approaches. These teams used palmy bird, and they used ensemble approaches. And most of them use all the original 600 articles for training their classifier. This is another table that's ranking the scores when we calculate the entity plus the relation type. And please note that the round numbers and the team rankings change depending on which evaluation measures we highlight. This is another table that rank the rounds and the teams based on finding a relation between right entity and identify whether it's novel or not. Again, please note the round number and the T1 key change. And finally, when we consider all the elements, the correct pair of entities, the correct relation type, plus the relation novelty or novel or not, let's notice that the numbers are lower. In fact, notice that the GPT models are really bad for this evaluation. This table shows the team evaluation of subtask 2. The median and mean numbers were calculated using all the submitted rounds. It is clear that this task is more difficult. The team had more resources and could compute the NER and ID quickly and had better results on this subtask 2. We noticed that the other teams use public available APIs to retrieve the predicted entities. This table shows ranking of the scores when we evaluate entity pair plus relation type. Unlike the subtask ones, the ranks of their best rounds differs in different evaluation metrics. We notice that in subtask 2, almost all the participants, their best rounds are the same among all different evaluation metrics. And these tables rank the rounds and the teams based on finding the relations between right entities and identify whether the pair is novel or not. And finally, when we consider all the elements, the performance is much lower. It shows that how difficult the end-to-end systems it is. And in fact, notice that the GD models are really bad for the end-to-end tasks as well. This scatter plot illustrates the precision and recall of all the teams for both subtasks. We will run in to discuss the teams in the red box. Here are the top performance teams. All of these teams used BERT, especially Palmy BERT is used by all the teams. But for the ensemble, one team used BioRED and one team used Beta, one team used biomedical bird. All the top performance teams rely on ensemble models, and these teams use logistic regressions to ensemble their models. And all of these teams report that the usage of Potato 2 API for subtask 2, and we see that their results are very similar. So now we already know what are the sense in this approach, and we will tell you what are the difference between them. These teams use data argumentations like GPT-4 to rewrite the input objects and use that for training models. These teams tell us that they incorporate large language models in their approach, and Team 156 use Llama tools with fine-tuning to improve their novelty predictions, and the Team 1.4 tools use GPT-4 for data augmentations. We look at the results from the participants and want to know what challenges their system faced. Were these challenges due to new topics like COVID-19, or because of different language? Sometimes the author used a more complicated language to express their ideas, their purpose. Sometimes diseases are caused by new species. Authors talk about the connection between the gene disease chemicals with the species, but not directly about the diseases. So, are these missing connections challenging case, or were tragic because entities were too far apart in the past, or they are not in the same sentence? It seems that the challenges were a mix of all these reasons. For example, we look at 53 objects related to the COVID, and out of these, 39 objects that had systems that could identify relations and with an F-score higher than 17%. This shows that even for topics not covered in the training set, the participant systems were quite good for figuring out the relationships. But we also have an example that was hard for systems which combine all the above issues. Here is an example of a difficult articles, and none of participants were able to predict the relations between the COVID-19 and variant and genes. However, all of them were able to recognize the binding relation between two proteins because they are in the same sentence. We believe that the reason could be that COVID-19 was only mentioned once at the beginning of the objects, and its relations with the variant and gene products was heavily implied or exploited in different ways. So let's talk about what are our next steps. The Coda Lab is now ready and updated with the BioRED BCA tested, and we are calling all current and new teams interested in testing their relationship methods to join us. And if you have any questions, please don't hesitate to reach out. We are truly thankful to all the participants from around the world, and your efforts have raised our BioRED benchmark performance to a new levels. And we would be very grateful if you could keep working on the new Coda Lab dataset. And we are also extremely grateful to all the 8 NLN curators. They dedicated almost all of their time to annotating the 400 objects in the test set, which took them approximately 6,000 hours. And we are thankful for the funding programs as well. Thank you for listening. We are now ready to take any questions you might have.